Given down below are all the studies in Correlation Engine database that are ranked based on its similarity to our list. Again, it's not only the direction of the fold change, but the magnitude of that change that factors into how well these different gene lists are correlated to each other. And it shouldn't come as a surprise that when we put our 130 genes in, we're getting a lot of papillary renal cell carcinoma studies. And if we actually open this top one here, we can see not only are we getting papillary renal cell carcinoma studies, but we're actually getting the original gene list that we used in our meta-analysis, which makes sense, obviously. But there's more information here. I mean, well, actually, let me take a closer look at this top one, just showing you how it's um, correlating to the different gene lists. I click on that. So in our list, we had 130 genes. They were all upregulated versus the control, or at least the sum of all that fold change was positive. If we look at the papillary renal, these type 2 versus type 1, we can see that about half the genes, there's 5,279 genes that change in this comparison. About half are up and half are down. But when we actually look and see what's common, all 130 are found in this list, number one. And number two is that all of the genes in ours went up, and they also went up in the type 2 versus type 1. So, and that is why his p-value is incredibly small. And I've been assured by people at Illumina, I'm looking at you, Jim Flynn, that you can actually calculate this. So again, it's not only, aren't we're not only getting validation that we can pull out papillary renal cell carcinoma studies from this cluster, but we're also seeing a ranking of how related it is based on these p-values. And what you can see is that the last one here is type 1 versus normal adjacent, and this is the least like our list. Then you could go up here in clear cell renal cell carcinoma versus normal adjacent. Now you're seeing it more like our list than it is to type 1. So again, this might tell me, just giving me biological context to our list, this might tell me that this cluster that we pulled out specifically because it is higher in the type 2 versus type 1 is potentially more like this clear cell carcinoma than it is to another papillary renal cell carcinoma type, the type 1. We can go down here, we can look, obviously these are some of the papillary renal cell carcinomas we used. But if we go down here, now we're starting to see other cancers come up that are related to ours. And if we look at some of these lists, we can see potentially why. Kidneys, obviously a lot of ductal tissue. Here it's saying that the ductal carcinoma is much more like our list than it is the lobular carcinoma. Again, these p-values are incredibly small. And what we can also see is that it's most like our list in the worst forms of that cancer compared to the less severe forms. Stage 3 to grade 1, or grade 3 to grade 1, stage 4 to stage 1. We can also start getting information on, some of the aspects of this cancer. Again, I mentioned that kidney cancer is predominantly males get it. Now we're seeing that if it's estrogen receptor negative compared to the positive forms of breast cancer, it seems to resemble our list, which is pretty cool. We can go down here and look at some of this, but again, this is giving you biological context. And that, say, if I found something that works on ductal carcinoma, it potentially might work on this form of type 2 papillary renal cell carcinoma. So again, more information. We can go down here. Here's another type of cancer that looks very similar to ours. So again, maybe in this type, if somebody figures out a good therapy for this, maybe we can apply this to our tissue as well, or our form of cancer as well. And again, we can go down here. I thought this was really interesting. Longitudinal analysis of progression to androgen independence. In this study, what they did was take prostate cancer cells and starve them of androgen or testosterone. I mentioned before that kidney cancer is predominantly males get this, and in fact, in papillary renal cell carcinoma, I read that Males are 5 times more likely to get this cancer than females. And if you look here, I mean, this might make sense. And this is telling you a lot that at 1 month, there's a lot of similarity that when you starve these prostate cells of androgen, all the genes that are going up in our study, or at least 108 of them, are going down in theirs. But what's interesting, and again, it's not just that one list, right, or the one study, is if we look that this effect might wear out about 11 months in, or definitely at 12 months. So again, this is giving you more information on your list. And what I really love about this is that it has nothing to do with what some experts said 20 years ago about a pathway that might not even be relevant. That what we are doing is taking our data and comparing it to the data that's out there. And in my opinion, I think this is kind of where bioinformatics is going, and that it's a more of a informative comparison than something that might be arbitrary as a old pathway. So that's curated studies. Now what we can do is we can take a look at body atlas. So let's do that.